Good day, everyone. Welcome. I'm Jack Van Horn from the University of Virginia, and I'm very proud to welcome you to this edition of our 2023-2024 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies, artificial intelligence, deep learning, and statistical modeling as they pertain to differing themes in the biomedical and health sciences. Our theme this year is focused on building partnerships for generative AI training in biomedical and clinical research, where every Friday we enjoy presentations from leading thinkers about the issues, promise, opportunities, and hurdles associated with the emergence of AI-based methods applied to biomedical science. Participants selected for our Biomedical Data Science Innovation Lab program leverage these seminar presentations as vital material for our culminating in-person manuscript and grant development workshop that we're holding in the Bahia Beachfront Resort located in sunny San Diego, California in June of 2024. We are very excited about this year's event. It's a very engaging program bringing together AI platform developers, biomedical research, and university level educators to better understand the power, promise, and potential pitfalls associated with the generative AI explosion. Today, I am delighted to welcome our speaker, Dr. Melissa Haendel from the University of Colorado Anschutz Medical Campus. Melissa is the Chief Research Informatics Officer at the University of Colorado Medical Campus and co-founder of the Monarch Initiative and the National COVID Cohort Collaborative, or N3C. Her background is in molecular genetics and developmental biology, as well as translational informatics, with a focus over the past decade on open science and semantic engineering. Her vision has been to weave together healthcare systems and basic science research and patient-generated data through the development of data integration technologies and innovative data capture strategies. Her research is focused on the integration of genotype-phenotype data to improve rare disease diagnosis and mechanistic discovery. She also leads and participates in many international standards organizations to support improved data sharing and utility worldwide. In continuing our 2023-2024 Biomedical Data Science Seminar Series theme, Melissa's lecture today is entitled Interoperability at All Scales, from Data Bits and Bytes to National Public Health Surveillance. She will note that making data reusable for discovery and shared analytics is laborious. Specific skills are required to make that task possible, and different perspectives are required to make it perform optimally. Equally challenging are the data reusers who function in a landscape of bespoke schemas, formats, and coding. Her presentation is going to cover all these different communities and their challenges and attempt to synthesize them into some areas of commonality. As always, we are streaming this lecture live and for recording via YouTube. And if you're watching on YouTube, thank you so very much for joining us. Also, our specially selected 2023-2024 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of Melissa's lecture. And with that, Melissa, welcome. We are so excited to have you here, and we are really looking forward to your presentation.
Thank you so much. It's really a great pleasure to be here. And I think especially exciting is the way in which I hope that this program will really stimulate and advance the idea that as we think about making more data reusable and interoperable, that we also think about the fact that all of those data sources that we might potentially use to advance public health, science, and any other societal challenges are always attached to people. And so interoperability and team science and open science with the people are just as important as the interoperability across the data sources. So with that, I will start with a term that my dear colleague Chris Shute termed the chasm of semantic despair, which is to say that when we think about basic research there over on the left and clinical care over on the right, that these are two communities that really don't talk to each other very much, and their data don't talk to each other very much. We have very different kinds of data and standards and tools that are utilized on the basic research side than we have over on the clinical side. And so one of our biggest challenges is really to think about how do we connect these dots in order to realize the dream of precision medicine and learning healthcare. And yes, that is me at the bottom of the chasm. So the sides of the chasm are not really so different from one another in the way that we manage data. So we have observations, we curate and code these data, and we try to integrate them. And so if you think about that through the lens of a clinician or veterinarian, we have patients or pets, we have documentation and electronic health record systems, we have clinical coders and coding systems like the International Classification of Diseases or SNOMED. And curators and integrators try to make sense of these data. Similarly, on the basic research side, we have biospecimens and model organisms. We document things in lab notebooks. We have biocurators. We have ontologies and vocabularies. And we have these knowledge bases and scientific biomedical databases. And so the question is really, how do we integrate these different worlds together so we can make the most of our biological understanding in the context of clinical care, and then also use the clinical data to help drive the best possible mechanistic and drug discoveries on the basic research side. And so the premise really is that semantics are the ultimate universal converter. And so really my talk today is all about all the ways and many different use cases that we can use semantic engineering techniques to help make data relate to one another to answer important questions. So first I'm going to talk about mapping the wilderness, curation rules, provenance, evidence, versioning, and attribution. And these are often words that are not terribly sexy in science, but fundamentally are these sort of undergird our ability to actually reuse data. And so I start off with this so that we can think, really deeply about where our data has come from in order to make it reusable and interoperable to address the important questions that we're aiming to work on. So this is just an example of a rare disease, Fanconi anemia. And this is just to show some examples of different types of mapping that occur across different resources. So for example, here at the top, we have Fanconi anemia B. This is a term that and a database entry that comes from the online Mendelian Inheritance of Man database. It has an identifier, definition, lots of literature is curated there. That's related to a term that has a different label in the Orphanet database. That's a European database. It's also related to a term from the NIH GUARD resource, which is a resource for patients. It's a database for patients. And then that's related to the medical subject headings, which is the terminology that's used to index Medline. And so, we have these different kinds of mappings. We also have different types of Fanconi anemia within OMIM. Some of these mappings are automated, but they're done by an automated tool that's not really reviewed by anyone. And so it's not clear what the validity of those mappings are. We have mappings that have to do with relating specific features, like an absent thumb, here represented by the human phenotype ontology, that's related to this subtype of Fanconi anemia A, which is has a genotype-phenotype relationship to FANC-A, a gene recorded in the OMIM record, but of course that's also related to the HGNC, the Human Nomenclature Committee's gene record, which is also related to the protein from UniProt. And this is just a tiny, tiny, tiny snippet of this semantic landscape of references between these different biological entities. Most of the time, these different entities are not well documented in terms of their specific relationships, making it really hard to compute across this landscape. So we've developed a standard for basically documenting the curation rules and the provenance for these different types of mappings called SESM, which is the simple standard for sharing ontology mappings standard. And in this standard, we can do something like, it's essentially a triple. You can say there's a subject, a predicate, and an object, where the subject would have the subject's label and identifier. So here, adenofibroma would have the identifier from the EFO ontology. We can have a predicate that says this is an exact match, which means that when we have different data sets that utilize this exact match, we can actually merge those records because we know has the exact same meaning. If you have a broader or narrower match, then you would not do that. And this is matched to the Mondo disease ontology for a term adenofibroma. And in this case, we can capture information about the mapping date, what our confidence was, whether it was human curated or automated. If it was automated, what method was utilized, who reviewed it, what model, what tools were used, and essentially on both a individual mapping basis as well as a whole data set file basis, we can actually declare what the mapping rules and tools were that were utilized, and we can version the whole thing. This makes that whole landscape that I showed on the prior slide extremely computable for merging and sharing data that's been mashed up in various kinds of ways. And so this is just showing a little bit more of that information, where we have information about the source of the mappings. We have our sort of short URIs, our queries, which are resolvable entities, and then, essentially an output in a table format that has all of the mappings. And these mappings are then all shared in a publicly available mapping commons on GitHub. So with that foundation, we're going to talk about reconciling the world's disease knowledge. So harmonizing terminologies across the disease community has been very, very challenging. There are very, very many different disease ontologies and terminologies, and there are different ones used by different communities, and both on the basic science and on the clinical side. They represent complex diseases, cancer, infectious diseases, rare diseases, Mendelian diseases, different countries, perspectives, and we needed a way to reconcile all of these different disease concepts and relate them to one another. And this was fundamentally how the SESAM standard was originally born. So why not just use the mappings that exist? We have cross-references or, just link outs across these resources. Well, these can be obviously used to crosswalk, but the problems is that they're often mutually inconsistent. There's n squared minus 1 sets of mappings, and they aren't really one-to-one equivalents. And so we found that essentially when we tried to bring all these different disease terminologies together, that we just had a big hairball and nothing was really computable. So for example, here are 3 different fragments of 3 different disease ontologies: the Orphanet disease ontology, the National Cancer Institute's thesaurus, and the disease ontology. And here we're looking at peroxisome biogenesis disorder and Zellweger syndrome. And you can see on the left in Orphanet that peroxisome biogenesis disorder has a subclass that's Zellweger syndrome, and it's a subtype of peroxisomal disease. Whereas over in the middle, Zellweger syndrome is a subtype of leukodystrophy directly. And then on the right, we have Zellweger syndrome is a subtype of peroxisomal disease. And all the different relationships between the sibling classes, the subclasses, and the parent classes vary across these different resources, as do the labels and the text definitions and their synonyms. So how can we determine if Zellweger syndrome is the same as peroxisome biogenesis disorder, or if it's actually the same as Zellweger syndrome? Because we cannot rely on the label. It's the definition that matters when a clinician is applying these terms. So we came up with a method called KBOOM, which is the Bayesian owl ontology merging algorithm developed by Chris Mungall. And what this does is it essentially is a logical and probabilistic inference model that takes advantage of the structure and the sort of priors within all of these different resources and many others not shown here, to generate a combined graph with a probability of equivalency for each set of nodes or cliques. And then those cliques with low probability where we have issues, a curator goes through and determines what's wrong. So for example, we found a whole branch of the medical subject headings mesh that was used in alpha numeric representation versus a Roman numeral. So we had diabetes type 2 with the number 2 and diabetes type 2 with the Roman numeral 2. And there was a whole branch that was duplicated in that way. So we were able to relate that back to NCBI, and they were able to fix it and merge those records. And now the Medline indices for those records are much more readily findable. And so this has been an iterative process and working with many, many different communities in order to make this happen. One of the really exciting things in having done this, and this was published several years ago, and we're about to submit an update for this, is that we were able to determine finally how many rare diseases are there actually. It turns out that in 1982, there was an estimate of approximately 7,500 rare diseases, and that is the number that is often quoted in many places, but we haven't updated that number since 1982. We took just 5 sources of diseases, NCIT, the disease ontology, GARD, Orphanet, and OMIM, And after doing this reconciliation, determined that just 5 sources comprise over 10,500 rare diseases that are unique. What's really interesting, though, isn't just the number, which is really not entirely determinable because we find new diseases all the time, but that only 330 shared disease concepts existed in all 5 sources. What this tells us is that we really need to reconcile the world's rare disease knowledge if we have any hope of having, of maximizing the possibility of a diagnosis, mechanism discovery, and drug repurposing for patients around the world. And so we really are keen to work with communities around the world, translate these resources into different languages so that everyone has access to the same knowledge. So this is what that looks like when you clean up the mess. So in the middle are the exact matches for that same term that I had shown earlier. This is adult refsum disease, which was on the graph earlier. And so these are all the different identifiers there in the middle in the different colors. And you can see the term identifier for the source synonyms as well. So in column A are all the different synonym texts, and we can see how those synonyms are referenced by the different terminology sources. And we can use that in the algorithm to help us determine equivalency. You can also see when we have references to non-equivalent terms over on the right. So we want to disentangle things that we've determined to be non-equivalent, even if they have the same label, or if they're just related diseases. And so this process has really helped us detangle this mess and make the overall landscape of any data that's attached to these terminologies, which are very many, UMLS, ICD. These are very commonly used disease resources, and we want to be able to integrate data from across those resources. And this is what that looks like when it's all cleaned up in terms of a hierarchy. We can now create that classification that leverages the world's knowledge from all these different sources in a much more holistic manner with the provenance, evidence, and attribution to all of the sources. So everybody wins. Okay, so now we're going to talk about harmonizing the anatomical diversity of life. This was one of my first sort of semantic people and data harmonization projects. So it goes way back. And in the beginning, we had very many different communities building anatomy ontologies in their little silos. We had the mouse phenotype ontology and anatomy ontology. We had the foundational model of anatomy in the human. We had developmental ontologies, and we have the gene ontology, which is very popular. And all of these different ontologies have representation of anatomy in them, and they have subclass relationships, partonomy, so it's anatomy, so part relationships, and developmental relationships, as well as other anatomical relationships. So can we just map across them? Same problem, right? Can we just say lung is a lung is a lung? Well, not so much, because it turns out that, we might want to take into account evolutionary relationships. So for example, the swim bladder has a homolog relationship to the modern lung. And how do we relate developmental stages to one another? So we needed a way to reconcile all these anatomy across metazoans. There's been a lot of effort to perform automated mappings, and this is an example that still exists today, 10 years later, where an automated lexical mapping was performed for things like looking at the zebrafish macula and the mouse macula, and that mapping is there, but that's not actually a useful mapping. Similarly, the human colon was mapped to the city of Cologne in Panama, also not a very useful mapping. So we need to make sure that, again, know where your mappings and your data came from and how you're actually going to use them. If we had this in a SESM file, we would be able to know that this was a lexical match that had not been curated, and we would have a lot more skepticism about utilizing this mapping. So coming back to the idea of data and people, we might ask ourselves, well, what does a famous dinosaur biologist and a craniofacial surgeon have in common? How do we build anatomy ontologies that can help both of these communities that seem so far away from each other? And of course, the answer here is the neural crest. So the neural crest is the developmental sort of patterning of the early embryo that creates the facial features. And it turned out that in some workshops that we had that included craniofacial surgeons and dinosaur biologists, there's some very interesting conversations to think about that developmental pathway and the needs of these different communities and understanding the biology and the clinical care for patients. And we actually came out with an incredibly robust representation by having these very diverse communities come together. This took many years to do this work. This essentially on the right is shown the timeline of some of the communities when they first started to work together to create what is now the Uberon ontology, which basically has kind of consolidated much in the same way we did with Mondo, but in earlier days with less tools and no modern AI technologies like LLMs. And so we were able to get these different communities to work together and create a shared ontological infrastructure by integrating those different ontologies and sort of de-siloing those different communities. It took a long time. So this is what we have now, and you can find out more about our project at uberon.org, which integrates many different ontologies and creates different slices for different purposes. So we can have a mammalian ontology derivative file. We can have a file that focuses only on the brain. We can have files that drive data collection forms. We can have files that can help validate gene expression and these sorts of things. And so this resource is really multi-purpose in reconciling anatomical knowledge across different scales and temporality and taxa. Okay, now on to interoperable phenotyping across clinical context, patient context, and model organisms. So phenotyping in this time is not so different from many, many years ago. And essentially, we still have these clinical notes that we can mine in various kinds of ways, but we don't really do a very good job of representing phenotypic characteristics in the clinical context. If it's not something important for billing or quality of care, or just a clinician tracking things from one visit to the next, it's not really found in the structured data. It's found in the notes, and the notes are easy to copy and paste these days. So we really struggle to get the right information out of the electronic health record, given that the nature of how the electronic health record is used and populated. The structured data that is provided in electronic health records is largely in the form of flat lists or taxonomies. So for example, problem lists or drug lists, or taxonomies like ICD, the International Classification of Diseases. And we really want to get to the point where we can actually represent information, knowledge, as a graph, similar to what I showed for Mondo or for Uberon, but we haven't really brought these technologies to modern medicine very much yet. So the human phenotype ontology really aims to represent phenotypic characteristics of a human being as an organism, the phenotypic characteristics that we could observe about an organism as a graph. So for example, we have terms like hyposmia or deeply set eyes. These aren't necessarily things, so deeply set eyes is not something that a clinician would necessarily put in a structured data field in the electronic health record, because it's not something that can be billed for or managed in terms of care. But it is something that's really, really useful when thinking about the biology of a human being. And also, because this is an ontology, we can create underlying axioms that relate that content to terms in other ontologies and other knowledge and data. So for example, that term hyposmia is related to the gene ontology term sensory perception of smell, and essentially having a lack of it. But that term from the go is annotated to 34,000 annotations in 22 different species. So all of a sudden, we can relate an enormous amount of mechanistic data from across the diversity of life to a single phenotypic term that might be recorded about an individual patient. So this lets us really think about how we can represent the biology of a patient in order to aid rare disease diagnostics. So here in the middle, we have what we've curated are these human phenotype ontology annotation profiles or phenotypic profiles. So here in the middle, we have Wiedemann-Steiner syndrome, which is curated to have a set of terms from the HPO like short toes, short middle phalanx of the finger, microcephaly, short stature, and the list can be several hundred long. And this is a case that that occurred in a colleague's clinic several years back, where within a few weeks of each other, a 14-year-old boy and a 3-year-old girl both came into the same clinic, were not diagnosable by standard tests. And it wasn't until the clinician went through the process of performing this HPO-based phenotyping to capture information like cone-shaped epiphysis of the phalanges of the hand, global developmental delay, thick upper lip vermilion. Again, not terms that you would find in the structured data of an electronic health record, but incredibly useful to represent the biological features of the patient, and allowed us to have these two patients diagnosed with the same disease, Weidemann-Steiner syndrome, but with a different variant in the same gene, KMT2A, and really kind of gets also after the idea of like some work that we do with the ClinGen community to understand when to lump and when to split a disease. So these particular patients have in some cases opposite phenotypes. So we have long toes in the boy, whereas we've curated short toes, or macrocephaly in the boy versus microcephaly in the gold standard annotation. And so, turns out that opposite phenotypes are very closely related into the graph. And of course, that actually allowed us to identify the causal gene here as being related to Weidemann-Steiner syndrome. So patients and families should be empowered to work as partners in this process. And the time is now for this. It takes a long time to capture that phenotypic information by clinicians and genetic counselors. There's care fragmentation, and we really finally are starting to have this knowledge democratization, which I talked about with the Mondo work, but also in many other contexts. And we have more tools and standards to help with this. So towards that end, we translated the human phenotype ontology into laypersonese. And so you can see an example here of, for example, small jaw would be the layperson term for a term micrognathia, or arachnodactyly would be spider fingers. And so patients can actually use the same term. So basically these are synonyms in the ontology that are that can be used in different patient-facing tools, where the identifier and the definitions and the structure of the graph are all the same, but the label that the patient sees would be something that would be more familiar to them. And so the question is then, how well can a patient do at describing their own features? And there are some features that patients would see in themselves or in their child that wouldn't necessarily be clinically identified. So for example, a baby who has inconsolable crying or doesn't have tears, or someone who snores, these are not necessarily clinically observable. And so patients definitely have a lot of content to contribute, and they're the best experts at their own condition. But not all of the human phenotype ontology is translatable into laypersonese. There are many technical terms, biochemical terms, that just don't translate very well into laypersonese. And it turns out it's about a third of the ontology is translatable. And so we did an experiment where we translated the HPO, and we created simulated profiles using the layperson terms. And if the layperson term did not exist for a term, we just rolled it up to the next one in the hierarchy. And we did this for all 7,500 or so of our curated profiles that I mentioned earlier. And then we created these simulated profiles. We also compared this to a survey instrument developed by Genome Connect, where we had mapped approximately 200 terms to the Genome Connect survey and did the same thing. And so you can kind of see there on the left where the full HPO clinical, at the time we did this experiment, had about 1,300 terms in it. The HPO Slim with the layperson terms was about a third of that, and then Genome Connect only had about 200. And what we found was really exciting is that when we used the layperson Slim version, could we actually do the experiment of doing that phenotype similarity algorithm that I showed you with Weidemann-Steiner syndrome? Could we recall the correct rare disease by doing that semantic similarity algorithm? And we were able to show both normal and noisy, because maybe the patient had a headache that day and they recorded that. So we tried adding noise and kind of randomly subtracting content and permuting what the layperson profiles might be in various ways to simulate more real life. And we found that even noisy versions of those profiles were able to correctly rank a fairly substantive number of the diseases within the top 5 hits. More than half of the diseases came back with purely based upon what a patient might theoretically be able to represent using the layperson terms. So this really tells us that patients could actually really be helping with this process, and that interoperability with patients can also be, a shared collaborative effort between the patient and the clinician to help improve the diagnostic odyssey. So now we'll talk about model organisms and why they matter to patients. So it turns out that that there's approximately 19,000 plus human coding genes, and only about a little over 4,000 of them have causal variations associated with them. If we look at the orthologs of those 19,000 plus genes in the top 5 most commonly utilized model organisms, worm, fly, zebrafish, mouse, and yeast, we actually have 83% coverage where we understand something about the causal effects of variations in those genes. If we combine them, we get 84% coverage of the human coding genome. So the inclusion of just 5 species boosts our phenotypic coverage of our understanding of the causal variation in humans by orthology by about 63%. So there's a lot of data out there in the model organism community that we might be able to utilize to help diagnose rare disease patients who might have variations in genes for which we don't know very much about. And so this is how we do that. We create a species neutral entity quality logic pattern. So here we might have hypolysinemia, which would be represented as a human phenotype with a quality of having a decreased amount of lysine in the blood. And so we have these semantic relationships that represent this term. And then we can represent a term in the mouse phenotype ontology with decreased circulating lysine level. It has a completely different label, but it has the same pattern of semantics in terms of its term representation. And this lets us reconcile the phenotypic relationships across species. And so what that results in is the ability to do the same kind of semantic similarity matching that I showed you for Wiedemann-Steiner syndrome, except here actually across species. And so now we can access that that model organism data that has that richer content and hopefully reveal candidate disease, genes for diagnosing patients with rare diseases. Okay, so now we'll go on to reconciling translational ontologies with clinical terminologies to realize public health surveillance for rare diseases. So one of the challenges is that we don't have rare disease definitions in the EHR right now. Most rare diseases do not have an ICD code, and therefore it's very hard to find rare disease patients in electronic health records settings. So how do we perform public health surveillance for rare disease patients if they're not, if they don't have a diagnosis of that rare disease anywhere in the structured data? And so the challenge really is that because there are so many of them, each different rare disease community really tries hard to get their rare disease to have an ICD code one at a time. And so we've been working with the WHO to really come up with a new strategy where we could take Mondo, which is that richer graph, and work together with ICD-11 as well as back populate for the currently implemented ICD-10 in what I call a Mad Max situation, where we can actually do a merger of the two so that we can have the more dynamic, regularly updated content in Mondo that's sort of more translational ontology with its full graph structure be sort of glued together with the more standard clinical terminologies that are used in electronic health record systems. And so I'd like to demonstrate this with a little bit of an example of how we're kind of approaching utilizing the electronic health record data for public health surveillance for rare diseases. And the reason this is so important is because many patients have a many years long diagnostic odyssey before finally recognizing that they may have a rare disease, before somebody finally recognizes that they may have a rare disease. And some rare diseases are potentially recognizable using electronic health record data, and we can screen for them, similar to how we would screen newborn screening for common genetic diseases. And so our goal is to try to do this for all electronic health record data, is to develop a strategy that for for especially the more common rare diseases, that we have computable phenotypes that help us identify those patients and get them moved along. And this is especially important in rural and disadvantaged communities that may not have access to primary diagnostic centers or academic medical centers, so that essentially the general practice clinics can actually help flag those patients and move them along a lot faster. So this example is about Fanconi anemia, which is the same disease I showed you in the graph in the beginning for SESAM, which is an autosomal recessive genetic disorder that is due to chromosomal breakage. And it's essentially a DNA mismatch repair disorder affecting 22 to 24 genes. And it has a variety of different clinical features: strabismus, low set ears, frequent infections, and leads to bone marrow failure and fairly extraordinary head and neck cancers, with patients living to be about 20 to 30 years old nowadays. And so it's a disease that is fairly readily identifiable at a very young age, but many of these patients go unnoticed until they get to a crisis stage when they're having bone marrow failure between 8 and 10 years old. So we'd really like to be able to find these patients in electronic health records before, so we can get them the care they need and get them ready for their bone marrow transplants and avoid the head and neck cancer. So shown over on the right is a set of HPO terms that we've curated for this disease. And so this has a Mondo identifier here at the top, and then all of the phenotypes shown here are readily accessible through the Monarch Initiative API or website with the link shown here. So you can kind of see how there's just a lot of different phenotypic features that are annotated to Fanconi anemia. You can see the synonyms that I talked about for Mondo up here in the text definition. So how do we figure out how to use this information in the context of an electronic health record? So if we have a lot of health records, we can have a potential suspected Fanconi anemia patient. We might have a clinical research data warehouse that uses the Odyssey standard OMOP, common data model, and concepts. So these are largely derived from SNOMED, but also a variety of other terminologies. And so these concepts take content from the electronic health record and map it to one of these OMOP concepts to help support observational research in the research data warehouses that you might find in your academic medical centers. A mapping file that uses SESAM that was developed by Tiffany Callahan as part of her thesis at the University of Colorado. She developed an OMOP to OBO translation mapping that maps these OMOP concepts to human phenotype ontology terms. And so you can see some examples here of short stature being mapped from SNOMED term to the human phenotype ontology term here. And these phenotypes would be associated with the Fanconi anemia Mondo term that I showed you on the prior slide and also in our website. So we can start to connect information coming out of the electronic health record to these human phenotype ontology terms, which also then gives us access to all the other data that might be linked to that HPO term. It's a little bit more complicated than that, however. So we also have information that requires semantic reasoning. So for example, there can be subsumption logic. We don't always have the exact term in the disease profile. So we need to do what we call fuzzy matching, like I showed you for Wiedemann-Steiner syndrome. We also map the LOINC terminology, which is a laboratory terminology of labs, for example, things like, individual lab that measures blood glucose, and then a reference range for what would be considered normal or abnormally high or abnormally low. And these are quantitative labs much of the time. And so we've mapped those in another mapping library called LOINC to HPO, which basically converts the LOINC term here, platelet in plasma by automated count, with this identifier. These are often have specific companies associated with the individual laboratory. And that those then can be combined with modifiers looking at the reference range to then here map it to a human phenotype ontology, thrombocytopenia. And so we have all of that mapping. And then we also have the notes. So remember from the very beginning, we talked about clinical notes being in the dark ages, and we want to extract if the clinician has actually mentioned any of these human phenotype ontology terms or anything related from these notes, and extract that information as well. So here we are looking for presence, microcephaly, and then that annotation would come out. And so then we hopefully can relate all this information to the Mondo disease ontology term. So I know you all have seen a prior lecture about the National COVID Cohort Collaborative, so I'm not going to spend much time on it today, but I definitely just wanted to highlight it here for those who are in attendance who have not heard about it. This is a very, very large publicly available electronic health record data set, is now the largest publicly available HIPAA limited data set in the United States, with over 21 million records, unique patient records from over 230 institutions. And these data have all been harmonized to that same OMOP model that I mentioned earlier. They represent, they're representative across the United States in terms of demographics, socioeconomic status, and health background. We spent enormous efforts harmonizing these data to that target OMOP model. The data is also linked to a variety of different other data sources. So we deduplicate records. So if a patient is seen at more than one location, we can reconcile those records. It's linked to viral variants, vaccine data, claims data, social determinants of health data, and about 80 plus other data sets. So there's a lot of integrations there. And we can use this data for public health surveillance to look at the effects of new variants in the context of COVID or the comparative effectiveness of drugs. And so what's really exciting about all this is it really took a village. There are over 450 organizations participating with approximately 5,000 users. And it really changed the way that we think about clinical informatics, because clinical informatics has historically been somewhat siloed because of the nature of not being able to share clinical data, and also just a somewhat competitive field. But in the context of this enclave that we created, which we call the N3C, we created governance processes and attribution policies that meant that whenever you shared an artifact, whether that was a new algorithm or a mapping file, that there was an opportunity for you to contribute to manuscripts that used those artifacts downstream. And this was created, of course, for the to address the pandemic. And we just snowballed the science, and the science just got better and went faster, with more people contributing in a team science way: epidemiologists, junior trainees, clinicians, machine learning experts, all working together to really kind of do the best possible observational research. So it's been a wonderful experience to really live through this process. And it's this foundation that I'm going to show you some really exciting new results that have been led by Brian Laraway and Evan Connolly in our lab, to actually use this data for the first time to kind of show how can we use all that mapping that I've just spent this whole hour talking about together in the context of electronic health record data to do that public health surveillance for rare disease, here, Fanconi anemia. So this is essentially taking all that data and trying to find who in the entire cohort of 21 million patients has looks like a Fanconi anemia patient based on those phenotypic features and all of those mappings. And so here we can see people that we think have FA. There's I think 27 or so patients in orange that have FA that are in the data set, and we can look to see which patients look most similar to those patients based on that same semantic similarity algorithm in the context of the enclave. So this is very exciting because it means now that we can take these individual patients that might be similar, especially these ones that are higher up on the semantic similarity score here, and we can rerun this algorithm over in the All of Us research programs researcher workbench, where we have genomic data, and we can say, okay, can we find any Fanconi anemia patients over there using this computable phenotype? And then see if they have variants in the known genes for FA to validate the computable phenotype. And then we can also run this computable phenotype locally in our own electronic health record data repository, especially at an individual location with one of our expert clinicians, where we know a lot of FA patients are seen, to see if we can actually identify those patients using the computable phenotype. And once we've done that, we will be able to run the same computable phenotype as the standard screening method to try to find FA patients in every electronic health record system. And we can do this for more and more rare diseases as we get better and better at this and have access to more data. So finally, I'd like to just share a few kinds of resources that some of the data scientists in the audience might be interested in utilizing. So this is the Monarch Knowledge Graph, and this has all of the gene to phenotype associations from approximately 126 species. There's causal and non-causal associations, and you can see many different data sources and types over here on the left, a list of all the different sources in the middle, the different ontologies and terminologies that we use to structure the data and integrate diseases, phenotypes, anatomy, genetics, and cellular anatomy and gene functions. And there's an API and data crunching, which is basically a bunch of different data reports that you can get to look at coverage in various ways. And we welcome requests, and we welcome contributors on GitHub. I also would be remiss if I didn't tell you about some of our really exciting LLM applications that we've been developing to speed our curation and management of these data. So the first one is the Monarch Phenomics Assistant, and we have a preprint out by Sean O'Neill in our group, and there's a link there. And this is basically a chat bot to query our knowledge graph. And so it's really exciting. So you can say, ask it questions like, give me all the genes that are associated with Fanconi anemia, or all the phenotypes that are associated with Fanconi anemia. So it's a really neat way of interacting with our API. We have a tool that interprets gene lists called Talisman, and that's also available in Archive. We have a tool that does parsing text to knowledge graphs called Spires, and our paper just came out this week by Harry Caulfield. So the link is there, and that's proven to be a really exciting way of sort of reducing the hallucinations that LLMs give you by using knowledge graphs and ontologies together with them. So if you're interested in new ways of kind of constraining LLMs in meaningful ways, that might be a great tool for you. We have a tool called Curate GPT, which helps find evidence in the literature more generally, and a tool called Mapper GPT, which maps ontology tools from the literature. And then finally, just wanted to talk a little bit about a new tool, Dragon AI, which can read GitHub tickets, documentation, background knowledge, and essentially create knowledge graphs and ontologies directly from all these different sources. And so it basically allows you, so if LLMs are general purpose text instruction processing machines, this tool allows us to preload the context that we want in order to help sort of train and direct the LLM to the things that we want, and then generate knowledge representations from those sources. And it can be from anywhere. It can be from your Google Drive. So we're pretty excited about this and look forward to having people test it out. So some takeaways, some common modeling for diseases, phenotypes, and anatomy facilitate interoperability across domains and around the world. We need all the organisms to understand biology and human health. The real world data is really messy, but if you harmonize it and you have a lot of it, you can reveal critical patterns, even with only 27 patients. And that good disease mappings and open science will save lives. I also want to highlight the idea that semantics can help us overcome the deluge of data. This is an article we wrote quite some time ago in New England Journal of Medicine on the left, called Classification, Ontology, and Precision Medicine, which is still very relevant today, which really talks about how do we go from the currently discrete clinical data that we're still capturing largely in the electronic health record, what drugs are prescribed, diagnostic imaging, those clinical labs I mentioned, to the world where we have a lot of emerging high throughput data. We have information about wearables and exercise and biomonitoring. We can study the microbiome. We can take pictures of our food and diet evaluations, and of course, all the multi-omics. How do we sort of think about the ways in which we might integrate those data together with those clinical data? And again, this is maybe a new kind of chasm of semantic despair. And I think we've done some interesting work, but there's so much more to do to think about sort of how you represent these kind of high throughput complex data sources in a semantic way as sort of a proxy. So for example, I could represent an abnormal EEG. I can describe those abnormalities using the human phenotype ontology, or I could have the raw EEG data. But I should be able to automatically derive the patterns from that EEG data to represent those phenotypic features in a way that's interoperable with those more discrete clinical data. And so this is one of the biggest challenges of our time in terms of precision medicine. So would like to spend some time thanking the enormity of people, especially Chris Mungle, Peter Robinson, and Chris Shute, who have been my partners on so many of these things, the Monarch Initiative and the N3C. But also all of the very many data sources and all the people attached to those data sources for which this work would not be possible, the way in which we integrate and work together with all these communities to harmonize data and build standards together. And then, of course, all of our funding from NHGRI, the Office of the Director, and from NCATS. And with that, I think I'm right on time to take some questions. Thanks, everybody.
Thank you, Melissa. That was great. It's so many different ontologies to kind of describe some different element of, something in this space. And I'm always kind of impressed with how ontologies themselves sort of spider out, they kind of grow arms and legs to kind of and capture more of what they're attempting to describe. And then they reach a point where that synthesis and that integration across them becomes necessary. And it just seems like one of those, in the enormity of that challenge and how you're bringing together all of this. It seems like it's a giant map where 1 mile equals 1 mile. And it really necessitates some of the tools that you've described here and some of the effort to go into it. And like you said, sometimes you get these Mad Max sort of mashups of these things. Can you describe a little bit more about some of the ways in which you kind of identify other ontologies that you decide are worthwhile in kind of looking for linkages? I mean, is there a criteria, or is it just, necessity being the mother of invention?
I definitely think necessity is the mother of invention. Sometimes, for example, if we have a small community or rare disease community or some other small community, they might have a very poorly structured terminology or ontology just because they don't necessarily have the background knowledge to do it well, but it might have incredibly specific content that we want to integrate. We found that a lot in the context of the anatomy work, for example, the biodiversity community and evolutionary biology communities. Other times we look for things where we know there's going to be a really critical need for interoperability. So for example, the Mondo ICD interoperability is really important because the impact of having integration and interoperability, could be quite large. We also look at the sort of quality of what is available. Is the ontology or terminology openly available? And I don't mean just openly available to use, but openly available to mash up and redistribute, right? So for example, some of those ontologies that I've mentioned do not have open licenses. So we actually started a project a number of years ago, not just not only for ontologies, but especially for data sources that might be encoded using ontologies, called reusable-data.org, to actually evaluate the redistributability of the resources. And that's actually a really nice way for students to look at that too. And I can put the link in the chat. It's reusable-data.org. It's a little GitHub file that is a little YAML file that you can sort of evaluate a resource. And what we found was that by evaluating resources that we wanted to have interoperable and redistributable that didn't have open licenses, that peer pressure of being evaluated in such a way actually got those resources to change the licenses to be more openly reusable and redistributable. And so that's a really important aspect, and important not to forget, because it's not really legal to just be taking these things any which way you want. And then finally, are there, is does it have an active user community? Are people making tickets? Can you see the discussions? Are there community calls or discussion forums? Because we want to make sure that when we're working on an interoperability effort, that we're supporting communities that are already actively trying to do similar things, and would be good contributors and good adopters of those interoperability efforts. So we especially like working with communities like that.
I was really glad to see you give Sean O'Neill a shout out, because he is a friend to our biomedical data science innovation lab activities. He joined us last year and gave us a little overview of the N3C. And you listed a number of different AI ontological kind of mapping and surfing tools. And those are all freely available and open source, I would imagine. And so our community should take a look at those.
Absolutely. And we would love people to try them out and contribute. They're all on GitHub, and the links are all in that slide.
Oh, that's fantastic. One other thing is, what is the obligation, the responsibility of the community who are developing the ontologies and the tools to traverse them in the context of education for the next generation of biomedical scientists, who presumably are going to be a little more facile than even the likes of you and I with, computers in the future. But passing on the knowledge about how to pull these things, how to search them, how to use these ontologies for, their research. What's that responsibility? What's it look like? And do you have any examples?
That's a really good question. I have to say there are very few locations and data science programs that have courses in semantic engineering or data standards. There's, I think a general, recalcitrance to thinking about standards as being sexy, and sort of, that what more do you need to know? There's the standard, right? But there's actually a lot of art in how you develop them, how you use them, and how you can use them together, clearly, subject of my talk today. And so I would love to see more courses like that. We have been leading a lot of efforts in the open biological ontologies community, or OBO community, and we have a community called a community education effort series. It's sort of self-learners called the OBO Academy. So that's freely available content for kind of educational modules, and it can be repurposed in the context of any kind of hackathon or educational program. So that has been one way that we've been able to disseminate to a broader audience some of our educational work around ontologies without having necessarily a face-to-face in-person class. But I would love to see some of that content, and any other content that anybody wants to contribute, be more readily utilized in the context of data science education. Seems like we unduly focus on the exciting AI technologies, our machine learning models, model reproducibility, but not so much on where did your data come from and how was it classified. And I think that there's a lot of innovation to be done in that space of looking at how the data is coded. And we talk about looking under the street lamp, because that's the data we have, and really kind of thinking about bias in a way that leverages those standards and where the data has come from and how it was captured in the first place. We also really strongly urge all trainees in data science to spend some time doing some kind of real world data capture activity, whether that be in the wet lab or out in the as a biologist in the field or in a clinic, to kind of really get after where that data came from and how it gets coded and structured in the way that you use it downstream. So similar to just knowing about ontologies is also knowing how they were applied in the first place.
Yeah, I agree. I think there are data scientists who are, in some of our training programs now, are very excited about all the AI stuff that you just mentioned. But it's like, an understanding and appreciation of where the data are born, I think they miss that. It's like magic happens, data appears, and oh, good, I get to apply a random forest. It's like
Right,
it's actually
exactly.
really helpful to know how the measurement device works, and how it functions and where it could misfunction, so that if you see something in your data, you don't immediately jump to some wrong conclusion. One final thing I wanted to
Exactly.
ask you about is that you mentioned a number of different conditions like Fanconi anemia and Wiedemann-Steiner syndrome. And I just heard on NPR not long ago about an effort to rename all North American birds so that they don't have the names of people. And I was curious if an ontology, the collection of ontologies like you've created here, might be able to undo some of this kind of eponymous, if that's the term, named after people kind of disease states, where we might actually combine things to identify new diseases that perhaps we've allowed ourselves to become kind of, I don't know, misguided by the fact that we have, Wiedemann-Steiner syndrome. Maybe it's similar to some other syndrome, and if we had known this, we could combine them based upon the features instead of the name of the person who defined it. Does that make sense?
Yeah, absolutely. And Fanconi anemia is a good example. So, one of the Fanconi anemia genes is a BRCA gene that causes breast cancer, right? And we didn't know for a very long time that those were related to one another, right? So, it's definitely, I mean, that's part of what the Mondo ontology is trying to do, is to reconcile all the different names. It's not so easy with disease names. So first of all, we never lose any names. We want to keep all those synonyms so that you can always find it. So even if Wiedemann-Steiner gets renamed to, KTM2TA related disease or whatever the gene name might be, you'd still be able to search for Wiedemann-Steiner syndrome. The problem is also about gene names, though. So if
Yep.
you, and we have a longstanding group between Monarch and ClinGen to discuss the sort of naming and lumping and splitting definitions for diseases. And it's challenging because gene names also change. So for example, I named a gene during graduate school axatrophin. The HGNC later changed that name to March 7, which you can imagine, as for the data scientists in the audience, what happens when you put a gene named March 7 in an Excel spreadsheet.
Yeah, that's right.
And so there was this whole effort to change all of the date gene names recently, a couple of years ago by HGNC, so that would stop happening. And somebody's done some analyses about like how much data science has happened based that is incorrect because of this Excel problem, right? So if we name diseases based on gene names and they change and they are ill-formed as per the dates, we're just punting that problem down the road. I think there's a lot of different personal opinions about whether we should get rid of all of the original, namers of these diseases. But at the same time, it's part of our history. These are the people who discovered them and studied them and cared for those patients. And they're still recognizable by those names. The gene-based names also are challenging because many of those are sort of combinations of gene-based names together with phenotypic features. And some diseases just have so many phenotypic features. If you try to include them all in the name, what are you really saying? Then you're back to having a disease profile, and it's not really a very effective name. So we need the names to be good handles, and we need lots of synonyms, and we need the provenance of the synonyms. But that's my opinion.
Yeah, well, it's a huge, big challenge, and we're going to need data scientists and ontologists and people, our next generation of biomedical scientists and data scientists to work together to kind of rectify some of these things and bring these knowledge spaces together in a way that you've pioneered here. So, Melissa, thank you so much for sharing this work with us. This was a delightful to have a chance to have a conversation with you. So thank you for sharing this. And again, your slides are available, and I've posted that on YouTube so people can find them. But thank you so much. We will be
Thank
we'll be taking a break for a couple of weeks, a couple of Fridays while we enjoy a spring break here at the University of Virginia and at other universities as well. But do catch up with us again at next time. In the meantime, have a wonderful Friday, and we'll see you